Hi, and welcome to Lambda U. My name is Gretchen Hubbard, and I am a Senior Solutions and Support Scientist at LICOR. In the previous course, we talked about blocking the membrane. This prevents nonspecific primary and secondary antibody binding. After the membrane is blocked, we are now ready to proceed with the primary antibody incubation step. Primary antibody incubation is critical in the Western blot process and can have significant impact on your results. In this figure, antibodies against the same target protein are compared. In lanes 2 and 4, there are more bands seen by Western blot than just the expected band at around 32 kilodaltons. These bands are often termed nonspecific because the antibody is recognizing and binding to these proteins when it should not. In lane 3, we see a single band at the expected molecular weight for the protein target, which indicates that this is specific for our protein of interest. Therefore, this antibody would be the preferred antibody for our Western blot assay. The choice of primary antibody for your Western blot will depend on the antigen you want to detect and the antibodies that are available that recognize and bind that antigen. There are several important selection criteria that should be considered for primary antibody selection. We will briefly review them here, but for more information, please refer to the Lambda U course on antibody selection. One of the first considerations you want to make for primary antibody selection is to find an antibody that is specific for your target protein. Another important consideration to make is whether the antibody is listed for your specific assay. An antibody that's listed for immunohistochemistry may not work for Western blotting. So you want to make sure the antibody is listed as working for Western blot applications. You want to pay close attention to the vendor website and review the peer-reviewed articles that are listed for the antibody. Make sure the antibody was used for a Western blot in the article, and confirm that the instructions on the vendor's website match the conditions used in the article. You also want to understand how the antibody was validated by the vendor. Antibodies can have lot-to-lot variability during production, so choosing a vendor that frequently tests antibody lots will help you ensure consistency in your Western blots. In addition, make sure that the antibody has been tested on whole cell lysate, not just purified protein. This will help test for specificity of the antibody. Making sure that the antibody has been tested on a wide range of samples, including cell lines and tissues, will make you more confident that the antibody will work well in your hands for your particular experiments. A lot of vendors now offer trial sizes of antibodies, which may be useful to ensure the antibody will work for your particular experiments. And lastly, be prepared to have to optimize the antibody you choose for your experiments. Using the proper positive and negative controls for validation in your own hands and optimizing conditions such as antibody concentration can ultimately help you perform a successful Western blot. In addition to the considerations I just mentioned, the antibody needs to be sensitive enough to give you the level of detection that you require. Reliability of an antibody will help ensure consistency in your experiments. Validated antibodies that have been used for Western blot and detect your species of interest will increase the probability of success in the assay. Looking at the antibody data sheet online and reading publications where the antibody was used for the Western blot assay will give you confidence that the antibody meets these considerations and can be successful for your research. In addition to these considerations, there are several different types of primary antibodies that can be selected for the Western blot assay. A target protein is often referred to as an antigen, abbreviated AG. Primary antibodies recognize and bind the antigen on the membrane through a region known as the antigen binding site, ABS. Sites on the antigen, known as epitopes, are where the primary antibodies specifically recognize and bind to form what's known as an antibody-antigen AB-AG complex. If the primary antibody is labeled with an enzyme, then it can be directly detected by the addition of the substrate. Or a labeled secondary will bind to the primary, and the protein of interest will be indirectly detected after the addition of the substrate. As compared to direct detection, indirect detection results in more HRP labels being present and ultimately leads to signal amplification. Indirect detection for Western blotting is the most frequently used method of detection. We will talk about 3 groups of primary antibodies today: monoclonal, polyclonal, and recombinant. Monoclonal antibodies represent a single antibody species that is made by identical B cells and bind the same epitope on the same antigen. These antibodies tend to be highly specific for the protein target and bind with very high affinity. These are good for quantitative assays because they have high purity and consistency, but they tend to be very expensive. Overall, monoclonal antibodies tend to result in the lowest background of primary antibodies, but they can also result in the lowest signal intensity due to only one antibody binding to one antigen. Polyclonal antibodies are a collection of antibodies that are made by different B cells and recognize multiple epitopes on the same antigen. In general, they give robust detection of target proteins because multiple antibodies can simultaneously detect multiple epitopes on the same antigen. This can, however, result in higher background. Despite this, polyclonal antibodies are frequently used for Western blotting because they are less expensive than monoclonal antibodies and still exhibit a high affinity for the antigen. Unlike monoclonal and polyclonal antibodies that are produced in animals, recombinant antibodies, known as RABs, are produced in vitro using DNA recombinant technology. Antibody genes are isolated and then incorporated into plasmid DNA vectors, and the resulting plasmids are transferred into expression hosts such as bacteria, yeast, or even mammalian cell lines. RABs are highly specific and circumvent some of the issues seen with monoclonal antibodies. RABs can be produced in a shorter amount of time, weeks versus months, are highly specific, and produce very low background. In addition, they do not elicit an immune response in humans and can be used as a therapeutic when fused to a toxin or drug. Unfortunately, the production of RABs requires a lot of expertise and money to begin production. Now that we have discussed the importance of choosing your primary antibody and the different types of primary antibodies that are frequently used for Western blotting, we will talk about the steps involved in primary antibody incubation with the membrane.